DNA replication. Why is that so important? So when we cover the mitosis and meiosis, and when we cover that the cell cycle, we pointed out the S phase, and we said that it's really important because this is the time when the cell duplicates its genetic material in order to the new cells would have a full set of DNA. So that's why was it so important. So the DNA has a 5' and a 3', and they are complementary of each other. So, and it has some origin of replication points where the replication will start. So it doesn't start at one point. Many places, so the replication will take shorter period of time. The two strands, the double helix, will separate from each other and form a replication bubble. The replication bubbles are along the DNA, and we have the 5', 3' ends. Okay, so it started to separate at a certain point, and we have an enzyme that starts to unzip the DNA, and it's called the helicase. It moves towards the right now, so unzipping the two strands, and the place where it's unzipping it, that's the replication fork. Now the red ones are the original strands, and we can say that the DNA replication is a semi-conservative method, that we will have one original in each double helix, and the other one is complementary of the original DNA. So we have the DNA polymerase that can increase the strand from 5' to 3'. So here is a nucleotide, let's assume, and this nucleotide has the 5 and the 3 ends, 5 and the 3 based on the carbons, and the DNA polymerase is going to match that base nucleotide with the original one and starts to build the nucleotides right after each other to the 3' end of the nucleotide. So the growing goes with the replication fork. This is called the leading strand. That was really easy. The other strand, of course, it's backwards. It will also have a DNA polymerase, but this DNA polymerase can only go from 5' to 3', so it will go towards the left, away from the replication fork. So that's really kind of the problem. Now, it's going to build small fragments on that strand. There's another DNA polymerase, and they will make short segments. These short segments are called the Okazaki fragments, and these fragments are going to be fused together by an enzyme. Okay, so this strand is going to be called the lagging strand. This is kind of a pain on the neck strand, but that's okay. So the two Okazaki fragments in here on this drawing is going to be fused together by a ligase, an enzyme called ligase. Ligase fusing together the Okazaki fragments on the lagging strand. So the DNA polymerase, what do we need to know about that? The polymerase is doing the adding of nucleotides to form the daughter DNA strands. Also, it has a proofreading capability. If something is not right, a wrong nucleotide is added to during synthesis, it will drop it out and just get another one. And also it repairs damage on the DNA due to UV radiation or toxic chemicals. Now, the important part in here, the replication that is a semi-conservative replication, the new DNA will have one strand, the original, and the new strand is the newly synthesized DNA.